Hi, welcome back to our channel. Now our topic is screening and confirmation of Vibrio parahaemolyticus by using FDA BAM method. Those who have not watched my part 1 and part 2 video, I have given the link below. Please watch, like, share it and subscribe my channel for regular updates. In my part 1 video, I have discussed about morphology of Vibrio parahaemolyticus. In part 2 video, enumeration, isolation and identification of Vibrio parahaemolyticus by using BAM method. Now this video we are going to discussing about screening and confirmation method of Vibrio parahaemolyticus by using ESFDA BAM method. Now let us start the video. Now Vibrio parahaemolyticus screening and confirmatory test. So these are the some of the screening methods: oxidase reagent, motility medium, gram staining test, and a urease test. So these are the some of the screening methods, and these are the confirmatory methods. If the screening is positive, then we will go for the confirmatory test by using Wagatsumagar Kanagawa phenomenon, or we can go for 3-tube MPN method. Now let us see what is oxidase reaction, oxidase reagent. Oxidase reagent transfer overnight cultures of T1N1 with the help of a metal loop into a filter paper containing saturated oxidase reagent. After transferring the overnight cultures, we can observe within 10 seconds on the filter paper, dark purple color will form on the filter paper, is called oxidase positive. Vibrio parahaemolyticus, Vibrio cholerae, Vibrio villinificus, Vibrio mimicus, all will come under oxidase positive. Few of the Vibrio parahaemolyticus strains are oxidase negative. Then next we will go for motility medium. Yes, motility medium. Vibrio parahaemolyticus and Vibrio cholerae, Vibrio species all are having polar flagellum. So single polar flagellum, the organisms are motile. We have to take one depression slide filled with broth, transfer the inoculum and place the cover slip on the top of the slide, view under microscope. We can observe the vibratory motility when we observe under the microscope. Then the gram stain. All Vibrio parahaemolyticus isolates should be tested for the presence of urease by either using urea broth supplemented with 2% NaCl or using API 20E kit. Clinical strains from west coast of US and from Asian countries have been predominantly urease positive. Urease production is correlated with the presence of TDH and TRH genes. Urease reaction is valuable screening test for pathogenicity for potentially pathogen strains. For urease test, prepare urea broth with 3% NaCl. Then inoculate heavy cultures into the urea broth. After inoculating into urea broth, we have to incubate 35 degrees centigrade plus or minus 2 degrees centigrade for 18 to 24 hours. After 18 to 24 hours, the urease production is determined by the pink color to the medium. So the pink color will develop. And the negative culture should be incubated for further 24 hours because of some of the Vibrio parahaemolyticus are so urease late reactions, slow urease production strains. Finally, when the colony is identified as Vibrio parahaemolyticus, then go for 3-tube MPN table for final enumeration of organisms, or we can go for Kanagawa reactions and Wagatsumagar. Confirmatory test. Wagatsumagar Kanagawa phenomenon. Yes. In 1968, the scientist Wagatsuma, who has developed a media to measuring the hemolysis capability of Vibrio parahaemolyticus, is called Wagatsumagar. Yes, this Wagatsumagar is nothing but high saline solution prepared with high saline means the Vagatsume agar contains 7% of sodium chloride plus D-mannitol is used as a carbohydrate for food for the Vibrio parahaemolyticus. So this medium was developed by using human blood or rabbit blood. So the hemolysis will occur after prepared Vagatsume plates. We have to transfer the inoculum from, or we have to transfer the overnight growth of T1N1 into the Vagatsume agar, streak onto the Vagatsume agar. So keep incubate at 18 to 24 hours at 35 plus or minus 2 degrees centigrade. So the hemolysis will occur. So the reaction, the Kanagawa reaction, nothing but the Kanagawa reaction. The Kanagawa reaction is correlated with the genes or proteins produced within the Vibrio parahaemolyticus are TDH genes and TRH, thermostable direct hemolysins and thermostable related hemolysins. Due to their pathogenicity, the hemolysis will exhibit. So Vibrio parahaemolyticus will exhibit beta hemolysis, means the colorless, means complete lysis. Lysis means rupturing of the cell or killing of the cell. Hemo means blood. Hemolysis means killing of RBC. Yes, complete hemolysis is called beta hemolysis. If alpha hemolysis, some strains like Staphylococcus or Streptococcus will exhibit alpha hemolysis, means incomplete, partially rupturing of the cells. So this is colorless and this is green color and this is no color, gamma hemolysis. So no rupturing of cells will occur. So the Vibrio parahaemolyticus will exhibit beta hemolysis on Vagatsume agar. So this is the Kanagawa phenomenon. Most of the strains which are isolated from humans, the samples which are isolated from humans, 100% Kanagawa positive. And most of the strains which are isolated from the estuarine environment are brackish water, 100% or 96 to 98% Kanagawa negative. And now the final is serology. Yeah, serology. Vibrio parahaemolyticus is a multi-serotype of organisms. Yes, there are about H antigens are there and K antigen is there, O antigen is there. H antigen, K antigen is a capsular antigen, O antigen also there. O antigen is covered by the K antigen, capsular antigen. Therefore, there are 76 recognized serotypes. Serologic tests by themselves are not used to identify Vibrio parahaemolyticus because of cross reaction with many other marine organisms will occur. Thank you for watching the video. If you like the video, click the like symbol, share it to your community and subscribe my channel for regular updates. Thank you and bye bye.